nice enough to come today. Maybe I'll come up here so I'm easier to hear. And yeah, it's great to resume the in-person Math Bio seminar. It's been a long and lonely past 2 years. We've managed to keep things going, but it's really great to finally get together again and be in person. So thanks again, Gabe, and looking forward to your talk.
All right, well, thank you very much for having me. I'm very glad to be here. This is the first time that I'm speaking about this work. So if you have any questions, any comments, I'd really welcome your feedback and any discussion during the presentation. Also, just quickly make sure that works. Of course, it doesn't. And excuse me on Zoom while I turn on my Bluetooth, which is on. All right, so great. So I'm going to tell you today about some work I've been doing thinking about the dynamics of stochastic integrating fire networks. So I'd like to start with a bit of a broad overview. And before I jump in, let me just ask, since we had all these technical difficulties, should I still aim to finish before 1?
Generally, let's go. Yeah.
All right, thanks.
Maybe we'll let people who need to leave.
I have to speak out.
Yeah, sure.
Yeah.
All right. Thanks. Great. So I'd like to start kind of talking a little bit broadly about the models that we use in mathematical or computational or theoretical neuroscience. So one kind of canonical model are the Hodgkin-Huxley equations, right? So this is a set of kind of really detailed biophysical equations governing the dynamics of the electrical potential across the cell at a particular location of some neuron, right? And these equations, right, have variables with biophysical meaning, right? Like the electrical potential across the cell membrane, the conductance associated to the flow of particular kinds of ions across that cell membrane, the reversal potential of those ions, right, governed by, or those currents governed by the concentrations of ions on each side of the cell membrane, and things like that. And of course, so we have Hodgkin-Huxley equation for the membrane potential, and also these conductances, the sodium, potassium conductances are also dynamical, and the joint dynamics of the electrical potential and those conductance gives rise to action potentials, right, in these neurons. So this model explains the generation of action potentials here, for example, in response to a short pulse of current applied at 5 milliseconds. So these are really powerful models. They have very rich dynamics. This is a model for a single neuron, right? If we want to use this to model the joint dynamics of many neurons, we might connect them up using synaptic conductances. And these days, with the advent of kind of large-scale electron microscopy, we're able to, by combining the Hodgkin-Huxley model with Wilfred Ball's cable theory, model the electrical potential right across the spatial extent of identified neurons in reconstructions of actual neural circuits, right? Not kind of simulated neural circuits, but actual ones, like in this little chunk of roughly 100 cubic microns of tissue from the brain of a mouse, from the IARPA Microns project. So we can combine these Hodgkin-Huxley equations with cable theory to develop really detailed biophysical simulations, right, of large neural populations. Those simulations, for me personally, have two big disadvantages, right? There are two main reasons why I don't personally do this work. One is that these models are very computationally intense, right? You have multiple ODEs, right? 4 ODEs in the Hodgkin-Huxley equations governing the electrical potential at every site in cell membrane. And if you want to model the electrical potential along the whole body of the cell, right, you have to divide it up into many compartments. So you have a kind of very fast explosion in the dimensionality of the model that you want to simulate. Along with that explosion in the dimensionality of the model come the two things I don't like, right? One is that it's hard to simulate on a computer, right? You need a bigger and bigger computer to do it. And the other is that it's harder to analyze mathematically, right? You just have a much more complicated model that's difficult to understand. So traditionally, what we've done in the field, right, to model large populations of neurons is instead to use highly simplified models, right? And this dates back to the 1960s with Steve Grossberg's work and later work in the 70s and onwards. Right? And these models can take a few different forms. Here's one canonical form of these models, where you have dynamics for something that we call maybe the membrane potential of a neuron or its activation. And that activation, right, or the membrane potential will be governed by a leak term, some input E, right, some synaptic coupling between neurons encoded in this matrix or operator J. And here that synaptic coupling will be mediated, right, by some nonlinearity phi applied to that activation variable. So that's supposed to capture somehow the mapping, right, from the membrane potential to the firing rate of the cell, or the number of spikes that it emits to communicate with its synaptic partners. So this function phi is basically, it's like a hyperparameter of this model, right? So we choose that function phi, and traditionally, right, we'll choose it to be some kind of sigmoid to mimic the fact that neurons, when they fire action potentials, right, each action potential takes a finite amount of time, right, a few milliseconds, and so a neuron can't fire arbitrarily quickly. There's, of course, another canonical form of these equations, right? The more traditional kind of Wilson-Cowan form, where we place that nonlinearity phi outside of the resting potential or baseline drive E and the synaptic coupling. And here we would interpret V as maybe itself being firing rate of the cells or the amount of synaptic drive, the synaptic current to them, something like that. Both of these kinds of models, right, are a bit more abstract than the Hodgson-Huxley equations, right? We don't directly have conductances governing the membrane potentials. We don't have a mechanism for generating action potentials or even action potentials at all, right, in these models, right? We have these nonlinearities, this nonlinearity phi, right, wherever we apply it. And that's supposed to capture something about spikes or action potentials. What these models are, actually, if you consider them as a mean field theory of an underlying microscopic model, is a mean field theory for binary switching neurons, where you take each neuron in your network and you say it has two states, call them 0 and 1, right, off or on, and it switches between those states stochastically. It's like a telegraph process, right? If a neuron is off, it'll stay off until it stochastically switches on, right, as a Markov process. And the probability that it switches on, right, is governed by this function phi, right, phi of the input that it receives from the network. Once a neuron switches on, it stays on, right, until it stochastically turns off, right? Again, as a Markov process here with some characteristic fixed constant rate that we've taken to be 1.
Can I just ask you a question about that?
Yeah.
Is there a difference between thinking of a whole bunch of neurons at half their maximum firing rate versus 50% at 1 and 50% at 0? Because you could think of phi as actually being a rate which is a continuous variable for each, rather than having binary.
Yeah, so you could make this mapping in kind of a few different ways. You could make this, you could have a network process V here, which is a mean field theory of a network of these microscopic switching neurons, or you could say that V is like the average activation across a population of neurons. There are a few different ways you can get there. But what we throw away in going from the microscopic stochastic model to this purely deterministic mean field theory is all information about the shape of the distribution of the activities in the neurons, right? We throw away exactly that kind of information. And we only capture something about their average activation. So, right, so we use these models. And what do we use them for, right? Well, they're dynamical models, so we use them to study the dynamics, right, of large populations of switching neurons. So one thing we might ask is how the activity in these mean field models depends on the strength of coupling in the population. And here you see that as we increase the strength of coupling in a single population model, right, the activity increases until with kind of strong enough activity, once we get to around j equals 1, so strong enough coupling, our activity is basically saturated, right, at the maximal allowed activity by this function phi that we specified here. Right? So here 1 is the upper bound for activity that we've imposed on our model. We have stable fixed points, right, all the way up there. Those fixed points are stable because of our choice of phi. If we take a corresponding linear model, right, where we just place phi by the identity map, once j crosses the critical value of 1, the activity actually loses stability, right, and explodes. So we have no stable fixed point for the activity when j is bigger than 1 in a corresponding linear model. So this stability is basically inherited from our choice of this nonlinearity phi, which is something that we just imposed on our model. So what do these models, these abstract models, tell us about brains, real brains? I'll give one kind of recent example of a use of these models in neuroscience that I'm fond of. And that's the description of an inhibitory stabilized network state. Oh, got a bit excited there. All right, so here, we'll consider a model with two populations, excitatory cells and inhibitory cells, and two input pathways to the network, right? We have kind of a main feed-forward stimulus pathway, the center pathway here. This is a model.
Can I ask a quick question?
Great, yeah.
Yeah. My question is, like, earlier you mentioned that your model can describing the stochastic process that it turned on with a probability that's related to the transfer function. But my question is, like, what if the transfer function is some function without upper bound, like a rectifier function, or
yeah,
or a superlinear function that Ken Miller is used a lot? So what in that kind of transfer function, how you interpret it into a probability?
So you would interpret these variables the same way, right? No matter what choice you make for phi, right? So phi is just a function that we choose, right? Like a logistic function here. And the interpretation of these variables is basically determined by the form of these equations. If you don't take a saturating nonlinearity here, right, then you expose your model to instabilities, right? Like this one on the right, right, where when the coupling strength J hits +1, the existence of a fixed, you lose the existence of a stable fixed point for this model, right? So the activity will explode, right? It'll diverge to either positive infinity or perhaps negative infinity if you don't rectify it.
Okay, thank you.
Yeah, good question. Thanks. So, right, what do we use these models for? One thing we used them for recently is to describe the existence of an inhibitory stabilized network state in the brain. Right, so this is a model of visual surround suppression. The idea being you have for a patch of the visual cortex, stimulus input it receives due to visual stimuli in the center of the neuron's receptive fields and kind of modulatory input from stimuli outside the center of the receptive fields, surround inputs. And in this model, we'll hypothesize that the surround pathway preferentially targets the inhibitory cells. And if the structure of this model obeys a couple of conditions, if the excitatory coupling is strong enough and you have enough stabilizing inhibition, it will have null clines for the activity variables that look like this. You'll have this kind of cubic shaped null cline for the excitatory activity and the sigmoid null cline for the inhibitory activity. And with perhaps a fixed point, right, on this increasing branch of the excitatory cline. And we'll call this an inhibitory stabilized, an ISN model, ISN inhibitory stabilized network model, because the stability of this fixed point is governed by the presence of inhibition in the model. So you have a condition, right, that the linearization of this of these dynamics at this fixed point has an excitatory element that's unstable. So the excitatory network, if it had a fixed point at this location, would be unstable in the absence of inhibition. So that's what defines this inhibitory stabilized network state. In that state, if we apply a surround stimulus, so if we apply an input to the inhibitory cells, that has the effect of shifting their null line upwards and to the left. And that shift of the inhibitory null line paradoxically leads to a reduction in activity for both the excitatory and inhibitory cells. So we stimulate the inhibitory cells, but find a reduction in their steady state activity. This kind of paradoxical response to inhibitory stimulation doesn't occur in a non-inhibitory stabilized network where the null lines have a different structure, so that shifting the inhibitory null line leads to increased activity in the inhibitory cells. We can see this paradoxical response in a simple little temporal simulation of the model, right? So we apply a little kick to the inputs of the inhibitory cells at 250 milliseconds here. And we see a transient increase in the prime rates followed by a decrease in their equilibrium activation.
But just to say something, what I see is a paradox. The new steady state, the inhibitory cells are actually getting less excitation. So you might think that's reasonable. But it's more that the excitatory cells in the new steady state are getting less inhibited but have a lower rate because they don't balance the bunch, which is kind of might seem as a paradox.
Yeah, so you could view this paradox multiple ways. You could say, well, I stimulated the inhibitory cells, but they ended up with lower firing rates. Or you could say, well, when I stimulated the inhibitory cells, right, I increased their activity. So I inhibited the excitatory cells in turn, right? So I lost recurrent excitatory drive for the whole network. That led to me having less inhibitory activity, right? But somehow I still stay with, end up with less excitatory activity rather than having kind of a rebound in response to that loss of inhibition. Sure. All right. So a couple years ago now, Alessandro Sanzeni, along with Nicola Brunel and Mark Histed and others, set out to test the predictions of this model in a really nice direct fashion. So they took advantage of genetically engineered mice that express channel rhodopsin, right, a bacterial protein that's ion channels that are sensitive to light in inhibitory cells. So they could shine light on a mouse's brain and stimulate the inhibitory cells. And then they also stuck in extracellular electrodes to record the activity of excitatory or inhibitory cells. And they found something that looked basically exactly like the prediction of this model, right? A transient increase in inhibitory activation followed by a steady state decrease in both inhibitory and excitatory activity. So they took this to be a confirmation, right, of the existence of this inhibitory stabilized network state in at least the mouse brain.
Do you know if this is in V1?
This plot is from V1. They also looked at other areas. They found it everywhere they looked. So this is something that we found from a prediction that we made using these highly simplified kind of neural field models, right? That seems to have been borne out, right? And this prediction, right, was that if the network is inhibitory stabilized, right, if it requires this recurrent inhibition to stabilize the excitatory activity, we'll see that requirement in this paradoxical response, right? And we see that paradoxical response. So the interpretation that I and I think most people in the field have drawn is that the cortex needs inhibition to stabilize its activity. That's a conclusion that we draw from this highly simplified, right, non-biophysical model. Right? So does that depend on the biophysics of the model, right? On the dynamics of that underlying model? And I'd like to say that it does. So to study this question, we're going to back off a bit and study a new model that I haven't talked about yet. It's not a new model in the field, of course. Other people have described it and used it. The first mention I've found is from Wolfgang Gerstner in '95. Philippe Robert and Jonathan Kiboul did some nice work on this model a few years ago. So here in discrete time, we have the membrane potential of a particular neuron at a particular time band, and it evolves with a leak term, some resting potential or baseline drive, some synaptic coupling between the neurons, and encoded in this operator, discrete operator J, that's driven by spikes, right, Dn, generated by these neurons as a Bernoulli process with spike probability governed by some function f that we impose on our model. So here f is, we can call it a hazard function in analogy with removal theory. right? So this is the probability that a neuron will emit a spike in a particular time bin as a function of its current membrane potential. So here, right, we'll typically take F to be a non-saturating function, right? So that if the subthreshold membrane potential of this model is large enough, the probability of a spike in a particular time bin increases and increases. So we can't have like our membrane potential go to 100 millivolts, but no spikes get generated. That would be silly. And we'll incorporate a threshold value in this nonlinearity here, theta, so that we have no spike probability until the membrane potential reaches this threshold value. And then once we reach that threshold, we have some probability of spiking, right, governed by the membrane potential. And here that probability is just a simple linear function. This is the simplest kind of spike emission or hazard function that we could, I think, use. When a spike occurs, right, we'll reset our membrane potential to some fixed value. So here we're modeling in the classic integrated fire style, right, the kind of Hodgkin-Huxley mechanism of action potential generation, that we generate an action potential when the membrane potential becomes depolarized enough. And when an action potential finishes, the membrane potential has reached some fixed value. That's kind of a cartoon of Hodgkin-Huxley spikes, but it's a decent cartoon. And here, right, we reset to within order dt of this value r, right, and the error is governed by that other term in the middle. So what does the activity in this model look like, and how does it compare? Yeah.
Just in the previous slide, I was wondering about the S to J.
Yeah, so here you could have some time delay in the coupling, right? So in the continuous time limit, this would correspond to a temporal convolution of some coupling filter with the presynaptic spike train.
This is not a function of the activity, right? This is just the delay actually prescribed, right?
Yeah, it could be a delay or some temporal shape to the synaptic coupling. Absolutely.
So differential equation.
Either, yeah, you could implement a differential equation on that, or you could say that Jij, the ijth element of J, is some function of the time S. And that's what we'll do in the continuous time.
What is I in this function?
Sorry?
What is I in this function?
So here I refers to the neuron, is an index for the neuron,
Oh,
and T is the time bin. So the kind of first thing that people did with this model is say, well, this reset, right, is this kind of nonlinear complicated thing. Let's replace it with a linear approximation, right? Instead, whenever a spike occurs, here we've non-dimensionalized, taking, right, the time constant to 1 and the reset value r to 0. Here, whenever a spike occurs, n dot, right, just decrease the membrane potential by a fixed amount. And if we look at traces, right, of our full kind of stochastic model here, and a model of this linear reset with linear resets where we force spikes to occur at the same time, we see that they have pretty different, right, looking dynamics. So I'm not very satisfied with this approximation. So to study this model, we'll build up the joint density functional of the activity across our across the neurons in our network and across the time of our simulation. So we'll start off by building up a conditional joint density of the membrane potentials V and the spikes N, conditioned on that Bernoulli spike generation. So we'll take a fixed sample of the spikes of the values of those Bernoulli random variables across neurons, right, and time bins. And conditioned on that, our joint density, right, is just a product of Dirac masses, right, enforcing the dynamics of our model, and that our spikes take on the sampled values. And then we're going to kind of take a few pages out of the statistical physicist playbook. We're going to introduce the Fourier representation of these delta functions, marginalize over samples of eta of these Bernoulli samples, and for convenience, we'll also take a continuous time limit. And in that case, we can write the joint density functional now of the membrane potential and spike train processes, right, in this exponential form, where the exponent we'll call the action, again, borrowing that language from physics, has the following form, right? So we have these auxiliary variables v tilde and n tilde. These arise from the Fourier representation of our delta functions, right? These are the Fourier transform variables. So we have a delta function enforcing the dynamics of our model here. Here we have that temporal convolution, Jij star n dot j that we talked about before. We have, and we have our resets here, n dot v. And then the last terms here, right, model the now in the continuous time limit Poisson spike generation of our neurons. So, and we'll use this an inner product notation to denote the integrals over time, sums over neurons here. All right, so we have this representation of the joint density functional of our model. So what does it give us? So the first thing that it'll give us is, which we'll see, is a really simple mean field theory for the activity of these neurons. So like to focus right first on these response variables or auxiliary variables, v tilde and n tilde. Right, these arose from the Fourier representation of our delta functions. That Fourier representation was bilinear, right, in say v tilde and v. So these terms that are linear in our auxiliary variables, they're like delta functions. This we can recognize as the cumulant generating function for the Poisson spike generation. This has, right, if we expand this exponential, it has a linear term, right, n tilde f, negative n tilde f, and it has a bunch of higher order terms, right? So if we think of our model, right, as having some distribution, right, in the membrane potential and spike trains, if we truncate all of this, the second and higher order terms in our response variables from this action, we're basically replacing that distribution with a delta mass, right, a delta function at some value, right, at the value that where v satisfies this equation and n dot satisfies n dot equals f of v. So this defines a purely deterministic or first order or classical mean field theory for our network. And it's a mean field theory in the sense that we're describing our model by fields, right? The field here, field refers to the membrane potentials v and spike trains n, the functions of time over fields. We're describing our fields with a distribution that only has a mean, right? It has no variance, it has no higher order cumulants. So that's the sense in which this is a mean field theory. And we can compare this equation, this simple equation, to the kind of classic models that we saw before, and there are two main differences, right? So the first difference is that we have this spike probability function or this hazard function f instead of that sigmoid function phi. And this function f, right, has an interpretation in our model. The other, of course, difference, right, is the presence of this nonlinear leap term, negative v times f of v, right? And this is the mean field version of our spike resets, right? So this is the dynamics that spike resetting gives us in our model. So if we return to the simple case of an uncoupled neuron here, right, we can write down also, right, the corresponding mean field theories here. So here we have, right, no synaptic coupling, but we have our nonlinear spike resets. And in the linear reset model, right, we just have this kind of reset dynamics, negative f of v, right, the negative firing rate. So we can solve for equilibria, right, of these mean field theories and ask how well they predict the firing rates of our cell. So in the blue, you have the firing rate of the linear reset model. In the dashed black line, you have the equilibrium of this first order mean field theory. The circles are the results of simulations of this model. And right, these solutions of the mean field theories have a very nice simple forms. And you see that there's, right, our linear reset model has a linear fi curve, right? It's linear in the resting potential or drive E, whereas the model with spike resets, properly accounted for, at least in that deterministic sense, right, has a nonlinear fi curve. We can also, as you might guess from the existence of this solid curve, compute the firing rate exactly by leveraging results from renewal theory, which we'll return to later. All right, so here we're talking about one neuron. And when we developed this density functional, we treated the synaptic coupling J as a fixed parameter of our model, right? We had some matrix of coupling filters J, and the parameter of our model determines the shape of this density function. We might also consider it, the coupling matrix J, as a quenched disorder in our model, right? We might say, well, we don't have direct experimental access to that, right? If we want to model experiments where we're just recording neural activity, right? Maybe we know something about its statistics, but we don't know actually the coupling between every identified pair of neurons that we'd like to model the activity of. So here we'll take a very simple choice for the distribution of elements of J, right? We'll say that they have some mean that scales like 1 over the size of the network N, and their higher cumulants are negligible compared to that. In the limit of a large network, right, we can compute the joint density of the activity, right, the membrane potentials and spike trains, by marginalizing out the coupling J. And what we find in the large N limit is that that density factorizes, so that our network basically decouples into N independent neurons, each neuron receiving a self-consistent mean input here, J, which is determined by the strength of this average connectivity J, and the average activity, the average spike train in that. So this isn't a totally new mean field theory, right? Philippe Robert and Jonathan Duboul described pretty much the same mean field theory and derived it in a different, more rigorous fashion back in 2016. Something we can note about this, right, is that this is a renewal process. This is the density functional of a renewal process, right? Every time a spike occurs, the memory potential is reset to a fixed value from which it has the same dynamics, right, that govern the spike emission probability. After a spike occurs, right, we just have first order dynamics. We can solve them if n dot is constant in time, right? If we have like an asynchronous state in the network. And that allows us to leverage classic results from renewal theory to calculate the average interspike interval of our network, which defines the average firing rate. The average firing rate is 1 over the average interspike interval. And it's composed of 2 terms, right? We have the time that it takes for the memory potential to reach the firing threshold, where it has a chance of generating a spike. That's the same every time, right? And then we have this term, right? This is the mean time the first spike after the memory potential reaches threshold for that threshold linear hazard function. And so the form of this, right, will depend on what hazard function we use for our neurons, right? And that's how we calculate the exact firing rate of a single neuron here in this example. We can also, right, use our mean field theory to look at networks of neurons, right? So we can, if we look at dynamics against the memory potential, we can infer the existence of a saddle node bifurcation for the mean field theory at some value of the coupling between 1/2 and 3. We can calculate the location of those bifurcations, right? There's one bifurcation that happens when the baseline drive E is 1 in the mean field theory. When it's super threshold, then the neurons will always be active because of that baseline drive. And for sub-threshold inputs, we have a saddle node bifurcation between just a quiescent state like this. We have one fixed point for the mean field memory potentials, which is below the spike threshold, so they don't spike, and the SPI-stable regime for the first order mean field theory, right, where you have a quiescent stable state and an active stable state. Right, and we can predict their locations, right, with, again, really nice, simple little equations, which is, to me, quite satisfying.
There's a question I'm confused about. So, you know, there's a lot of work on networks with random, zero, and Gaussian connections. So here, J is the mean, which is the 0 there, but I know they have a lot of interesting phenomena. And so I guess I'm thinking about scaling event because there's n
yes, exactly.
J goes to 0, but the coupling goes to the square root event. So where is that in here?
So here we took the coupling. So our assumption that the mean connectivity was 1 over n, but higher order statistics were like at least 1 over n squared. That's consistent with a model where the individual synaptic weights all individually scale as 1 over n.
Okay. So it's just 1 over square root of n.
Yeah, so if you introduce 1 over square root of n, that's started to look at that, right? You get kind of a more interesting mean field theory with internally generated fluctuations also you need to determine the statistics of self-consistently. So you have that same kind of thing shows up in these networks also, but with spike reset dynamics. All right, so we have here that bifurcation, right? Of course, these are bifurcations in this purely deterministic approximation of our model, right? That's a pretty coarse approximation that we've made. In simulations, right, we see that it seems to describe things pretty well, right? At least in each of these regions, and fairly close to this bifurcation between monostable low activity and bistable bistability in the mean field theory. We have and bistability, right? We can turn on activity here in D with an input pulse and turn it back off again. And we can also leverage those exact firing rates from renewal theory to calculate exact bifurcation diagrams for our model. And we see that the first order deterministic model dash captures the locations of these bifurcations pretty well, right? It doesn't compute the actual firing rates very well, but it gives us a decent idea of where bifurcations happen.
So you never destabilize the quiescent state.
Yeah, there's always a stable quiescent state, and I think you showed back in 2016 that in the full stochastic model, right, that's actually the only true attractor, right? Because once the network enters a quiescent state, it has no spikes, right? No way to escape it, right? But in the active state, the neuron is kind of generating spikes, generating fluctuations that after a long enough period of time, and it might be very long, right, as you showed, will kick the network into the quiescent state where it'll just stay.
I mean, I should read my paper again. But I kind of remember there was this threshold where, I mean, above which in the in-coupling, above which we could no more have the low activity regime. I mean, it depended on F, on the activation.
Ah, interesting.
So in your case, you always have this quiescent state.
Well, so we have this quiescent state because our hazard function is 0 until some threshold.
Right.
If it wasn't 0, right, then maybe we could lose this quiescent state.
Actually, because I think it was the derivative at 0. I mean, we look at power functions, If you have powers larger than 1, you're very flat. We maintained also quiescent state. For linear, I think it switched, and for supralinear, like square root, for instance, then we may lose the quiescent state because the activity buildup was fast enough to stabilize.
Yeah, interesting.
But
No, that's an interesting point. So here, right, we have this threshold linear transfer function, and I would, I think usually in the field people would use I guess for small inputs sublinear, right? Like power laws with powers at least 1. The idea being, right, that as the membrane potential increases, right, our membrane potential variable here, V, is supposed to be the subthreshold membrane potential. So you'd hope that as that increases, right, as the subthreshold membrane potential gets above -50 millivolts, -30 millivolts, right, you really should generate a spike, right? So you'd think that this hazard function should increase quickly for large membrane potentials rather than, more slowly the larger the membrane potential gets.
This is good.
It's quicker close to threshold, right? And yeah. But if you somehow get, far away from thresholds, then
for stability of the quiescent state.
It's that's a good point. Yeah, that's a good point. That stability of the quiescent state probably depends more on the ability
You
thresholds.
didn't change the slope of your threshold of the threshold linear.
I've only looked for slopes greater than 1. I didn't look at smaller ones. So, all right. So another advantage of this approach to determining a mean field theory is that it's really easily extensible, right? We talked about how we could extend it to models with stronger coupling, right? Synapses that scale like 1 over the root of n. We can also easily extend it to models with multiple populations, right? The simplest case being the network of excitatory and inhibitory cells, here with kind of global strengths for excitatory projections J and inhibitory projections GJ. And there we see kind of qualitatively similar dynamics in the mean field theory, as long as both populations receive the same baseline drive E, with now the presence of a maximal inhibitory strength for the existence of bistability. So if inhibition is too strong in this model, you won't be able to sustain an active state unless the input is superthreshold. Again, calculate really simply, right, the locations of those bifurcations as simple analytic functions of the primers. And again, we see, right, that this simple deterministic mean field theory gives us a pretty good description of the location of bifurcations in simulations and in numerical solutions of the exact triangle of the model. All right, so that's kind of equilibria. We talked before about, and equilibrium in excitatory inhibitory networks. We talked before about this inhibitory stabilized state, right? And it had two requirements, right? One, that at the fixed point, the network, the excitatory subnetwork should be unstable, but that the whole network, right, should have some stable, should be stable at that fixed point. So what does this look like in our model? So if we take a simple kind of classic mean field theory here, we see that as, right, the coupling gets stronger, we have the presence of an unstable fixed point here just above 1, right? So the activity will explode with this linear transfer function, right? So we didn't see this like back at the beginning of the talk because we imposed that function phi saturated, right? If we don't do that, then our activity can explode. But if we incorporate spike resets into the model, right? If we incorporate this spike reset term, we said that that's not actually the case, right? We have a stable fixed point for this single excitatory only subnetwork, no matter what J is, right? Its location, of course, depends on the coupling strength J, but not its existence. As long as we're right within that bistable regime. So, right, so we can see this, we can look at what happens if we take a network in such a state, right, and stimulate it. So we're taking our excitatory inhibitory network, and we're giving a little extra stimulation to the inhibitory cells. And we can see that if we do that, first we can observe a paradoxical reduction in the inhibitory firing rate here. And if we stimulate them again, we might actually shift the network into a really different state where the inhibitory cells are of even lower activity and there's no recurrent excitation. We've totally turned off the excitatory cells. Understand these. Whoa. Sorry about that. We can understand these behaviors, right, again, by using that simple deterministic mean field theory, right? We can look at the phase diagram of the inhibitory and excitatory mean field memory potentials. We can see that when the inhibitory input is weak enough, we have a stable fixed point, right, on an increasing branch of the excitatory null line. And as we increase the input to the inhibitory population, right, we shift that inhibitory null line up and to the left, or actually just up, right, so that at first we move that fixed point down this branch of the excitatory null line until we pass through the excitatory null line and lose that steady state with active excitatory cells. We can calculate, again, using this mean field theory, conditions for when we might observe this kind of paradoxical response to inhibitory stimulation, right? We need to be on this increasing branch of the excitatory null line, and we need to have a fixed point on it, which means that at threshold for the excitatory population, the inhibitory null line had better be below the excitatory null line, right? Below it there, above it at the inflection point at the top of the excitatory line. Those two conditions, right, give us these following expressions. They're not quite as clean as the other results we obtained from the mean field theory. They're a little bit longer, but they're still very simple functions. Yeah. All right. We can use these to calculate where we expect to see paradoxical responses and confirm that we see paradoxical responses in wide regions of the parameter space, even when the network has stable fixed points for the activity, without any inhibition, right, in the absence of inhibition. Right, we can do a lot more with this.
So
thank you all.
maybe let's give Gabe a round of applause. I suggest maybe we could move to either the lounge or another room for more discussion.
That sounds great. Thank you all. Thank you all on Zoom. If you have any questions on Zoom, we have to move now, but please feel free to email me.
We can reconnect.
Yeah. Okay, yeah, we'll try and reconnect.
Thank you.